So in the second part of today, we're gonna go through a little bit more details, but we're going to be covering a lot of the stuff we talked about in the first half. Again, okay? We're gonna add to what we covered a few more details, but we're gonna kind of review some of these concepts. So what I'd like to do here is, if you look at today, there's like 28 slides. That's it. You know, we're spending like 2 hours on 28 slides because I want to make sure that we cover it thoroughly. So there's a little bit of new stuff here from what we covered in the first part of the lecture, but much of it's going to be review. But we're going to fill in a few more details. So sticking with the theme of B cells, let's talk about this concept of clonal deletion. We talked about T cells. We talked about how they have central tolerance and they have peripheral tolerance. And central tolerance and peripheral tolerance are very good things, right? They're good things because what the body does, the body says, okay, I'm going to go through this clonal diversity, and I'm going to manufacture all sorts of different variations of T cells and B cells now. And by making all these different creative versions through clonal diversity, inevitably I'm going to make some that are self-reactive. But I don't care, because I'd rather make sure that I get as many variations as possible, and then I'm going to have some programs in place to get rid of the ones that are self-reactive. Some would say, well, that's a very inefficient process. Yes, that is. But the body says the immune benefit of being creative with the diversity of what I could create is worth the biological energy to get rid of many of the ones that I actually manufacture. Okay? So, for example, in B cells, when we go through clonal diversity, we now follow it with what we call clonal deletion. That's actually what central and peripheral tolerance is called with the T cells that we came from, clonal deletion. But in the B cells, remember the B cells from the bone marrow, they don't go to the thymus. They only have a central tolerance in the bone marrow. And they don't have a backup mechanism. They don't have a backup mechanism like what you saw with T cells. T cells have a backup mechanism known as what? The peripheral tolerance. B cells in the bone marrow only have central tolerance in the bone marrow. And if it's self-reactive, then they trigger it for apoptosis and they get to try again. Well, this happens so frequently that over 95% of the B cells that you create in the bone marrow are triggered for apoptosis. 95% or greater. Does that just blow your mind? So your body's saying, I'm going to spend all this biological energy, and 5% of the time I'm being successful. And that means I'm going to create non-self-reactive B cells. That's how important this is to our system. T cells have a backup mechanism. So instead of 95%, they're about 90%. 90% of the T cells that you make between central and peripheral tolerance get eliminated. Okay? But they have a backup mechanism of peripheral tolerance to catch some extra ones, so the numbers are, I guess, a little bit lower. A little bit more efficient process with T cells. Okay? Yeah, these are approximates, but I mean, bottom line is it's in the 90s. It's a little bit more aggressive for B cells because they don't have that backup mechanism. So 95% for B cells are signaled for apoptosis. And with T cells, it's about 90%. Okay. Now if we look at why apoptosis is so important, because if you catch it early, it's not as inefficient as it might seem. Because if you catch it early, before you proliferate, you can actually try to reprogram, and then you don't waste a lot of it. And it's in the bone marrow centrally, so it's actually not as wasteful as it could be if it was other places. Now, if the B cell actually becomes immunocompetent, and it comes across an antigen, it has activity in a couple of ways. So this was the slide before the break that talked about the valence, it talked about memory B cells, it had diagrams on the right side that showed opsonization and phagocytosis and neutralization. And this is the follow-up to that slide. So antibodies that are manufactured from these B cells, they can have direct or indirect effects. The direct effects are listed here. Neutralization, it binds to the toxin and it inactivates it. Agglutination is it starts to clump the antigen. That means the antigen is already insoluble, meaning it's a precipitate, and it starts to clump it in the blood, and then it sort of slows it down, prevents it from circulating, allows for a macrophage to come and help digest it away. Or the direct effect could be precipitation. If the antigen is soluble in the bloodstream, then the B cell antibody could bind to it and make it fall out of solution, precipitate it out, so that it can't circulate around. These are considered direct effects of how antibodies work. Now there are some more indirect ones. Opsonization, phagocytosis, and inflammation. So these pictures are associated with indirect effects. Specifically, oh, my arrow's not working. There it goes. Specifically, if we look at opsonization and phagocytosis, these are very similar to each other. Opsonization is a term where an antibody binds to a pathogen, and then that pathogen is marked. It's marked for ingestion and destruction. And very similarly, phagocytosis is the process of engulfing and digesting. So a lot of times opsonization and phagocytosis go hand in hand. It becomes opsonized, labeled, identified, marked for ingestion, and then it's phagocytosed up. Okay, and you can see this diagram here nice and clearly that the opsonized cell, the phagocyte engulfs it and then starts to digest it away. The third indirect effect is stimulating an inflammatory process. I explained at the beginning of the lecture that we're going to see how the adaptive immune system leverages our secondary line of defense, which is inflammation. And so why wouldn't you use inflammation if you have it available to you? And that's exactly what antibodies do. So you can see here with this inflammatory trigger, this is triggered by the complement cascade. So we've got our FC, our crystallizable receptor right here. You can see the antibody binding. You can see the complement comes in and helps to activate. And the complement products that we've studied before, specifically C3A and C5A, these actually help to recruit neutrophils and enzymes, and those guys come in and start manufacturing reactive oxygen species to chew up the antigen or the microbe, digest it away. You remember that Fenton reaction that takes place? And how you get these reactive oxygen species contained within a phagocytic cell? And so if you engulf an antigen and you fuse it with a phagolysosome, and you can create an internal Fenton reaction by cranking out reactive oxygen species and nitric oxide inside the confines of the cell, you can actually digest it away from inside of the cell. Pretty cool. You need these neutrophils to come into the area. That's an inflammatory process, and you know exactly how to describe neutrophil infiltration, monocyte infiltration, right? With the margination, with the activation, with the rolling and the tethering, adhesion, and then diapedesis across. You can explain all those steps, right? You can put them in the correct order. How they relate to an inflammatory process, And complement will trigger that to happen in the presence of the appropriate antibody. That's an indirect effect because it happens eventually, but it is an effect of B cells. Okay, lastly but not least, on the B cell category, and then we'll talk about more details on T cells, and then we're done. B cells create, after they've responded, memory B cells that stick around. These memory B cells are what are responsible for vaccinations historically. So the primary response is dominated by IgM, and it's followed by a relatively high IgG response that lasts about 3 to 5 days. But the next time you see it, and you trigger antibody production, the IgM levels stay about the same, but the IgG happens much faster, immediately. Doesn't take 4 to 5 days, or 3 to 5 days, excuse me. And it's approximately 4-fold higher than what it was initially. So there's an interesting story, which is sort of the basis of vaccinations. The first successful vaccination happened in 1796. Okay? This was a physician by the name of Dr. Jenner. Dr. Jenner was noticing, we were in a real farming era, this is before the Industrial Revolution, and he was noticing that, Dr. Edward Jenner noticed that young women who milked cows often got cowpox, but they rarely caught smallpox. Just an observation. So he thought maybe getting cowpox prevented you from getting smallpox. That was his hypothesis. And of course, you didn't have to fill out any forms, so he just decided to experiment on some patients. Right? Isn't this interesting? So to test this idea, he infected his patients, a subset of his patients, with cowpox on purpose. They developed memory B cells, and then he exposed them intentionally to smallpox. And they didn't get smallpox. He didn't know how it worked, he just knew that it worked, okay? And this was the beginning of vaccinations. In fact, vaccination comes from the Latin vacca, meaning cow. So it's cownation, is really what this is, okay? Dr. Jenner created vaccinations. And since 1979, I'm imagining most of you were born after that, so you have never been vaccinated for smallpox, because it was eradicated in 1979, meaning there was no longer any cases in the world, and so we consider the disease gone from the planet. Okay? Although, if you're not vaccinated for smallpox, you are at risk of contracting smallpox, and that's one of those concerns about biological warfare. Right? So smallpox vaccination is pretty obvious. Usually it's on the shoulder. Okay? It's more common in international individuals because it was eradicated in the U.S. earlier than it was in a lot of other nations. So a lot of times if you have friends that were born maybe in the 70s or maybe even the early 80s, or more like in the 70s, they were vaccinated for it. They have a little smallpox scar on their deltoid. Okay, cell-mediated. We're going back and forth between B and T cells today. Back to T cells, cell-mediated. The clonal diversity that takes place here, right, happens at the level of the thymus. The clonal diversity that happens for B cells happens where? In the bone marrow. Here it happens in the thymus. Thymopoietin. Thymopoietin is a hormone that's released by the thymus to help with the maturation process of T lymphocytes. In this process of central tolerance that we went over in great detail, I want to show you once again, just in a more summary type of slide. So this is a diagram of the thymus. Okay, it's a lobular structure. It sits right here about mid-sternum, okay, above the heart. And you can see that the T cell precursors come from the bone marrow. They proliferate, and as they proliferate, they go through this creative clonal diversity, generating all these different variants. And then they're screened in the thymus. And they're screened in the thymus, and many of them go through cell death. In fact, about 90% of them, many of them, 90% is the combo between central and peripheral. So many of them get screened here, and the idea is they exit out being immunocompetent into the periphery, but not self-reactive. But we know that in the thymus, sometimes we don't catch it, and so we have the exiting of cells. You can add in here. This is where peripheral tolerance picks up. And between the combination of central tolerance and peripheral tolerance, you clonally delete about 90% of your T cells that you make. 90% of the T cells that you make are eliminated at the cost, the biological cost, of being very clonally diverse, so that you have all these different variants that can react with many different types of things. Okay? So, the ones that sneak out, B cells or T cells, are going to be the basis for the next lecture series after the exam, when we talk about autoimmunity, and we talk about hypersensitivity, when we talk about things when they go wrong, and why we react to certain things that we shouldn't react to. It's usually because of a breakdown in these tolerance mechanisms, and you have self-reactive clones that are out in the circulation. And now you know where they come from. Yes?
So B cells, terminate the bone marrow, and so the T cells terminate the
Thymus.
Does that also mean that they know whether it can be a helper or a killer T cell or the thymus, or that's one thing?
Oh, the thymus is the point at which where it just determines if they're a cytotoxic T cell or if they're a natural killer cell, for example. But the B cells are determined in the bone marrow. Right. So their determination of B cell differentiation happens in the bone marrow. The T cell differentiation happens in the thymus.
And it's also where it decides what antigens it's going to be targeting?
That's correct. In the bone marrow, yeah, for the B cells.
Oh, okay.
T cells in the thymus, yeah, that's correct. There are subclasses of these T cells, but we're not going to get into that level of detail. So you're going to see like Th1 and Th2 nomenclature. Right? You'll see these subclasses that take place, but we don't go to that level of depth. Again, focus on what we're covering in lecture. Okay, to finish out the T cells, we want to look at how their activity occurs. And so I'm going back to these pictures that I told you we'd come back to. They should be familiar. We're going to stay on this particular image for a couple of slides. I've drawn in my own little somatic cell. What's a somatic cell? That's a nucleated cell that can be any body cell in the body. So a somatic cell that's nucleated that becomes infected could actually present the antigen. Does that make sense? So antigen-presenting cells are specialized antigen-presenting cells. But if you have an infected cell, it's going to display that infected antigen on its surface, and in essence is presenting an antigen. That's a somatic cell. So the picture and the book don't really describe it, but I drew it in myself, and that's what's being shown on this particular slide. So we've got dendritic cells that can present either to CD4 or CD8 cells. And then you have the somatic cells that only utilize the CD8 pathway that goes to the right. The dendritic cells present to CD4 positive cells, which is MHC class 2. They migrate into this lymph node. Sometimes they'll activate the cytotoxic side that we'll talk about here in a moment. Most oftentimes it's the T helper side. Now, we stay on this slide and we talk about the steps, and that's what's shown on the far left of this slide. Same image, just the steps that take place. So as the dendritic cell comes in, the activated CD4 positive T cell enters the circulation, migrates to the site where the antigen, right? So here it activates, migrates into the circulation, goes to the site where the antigen is, and it's going to cause a number of different possible activities. It can cause macrophages to become activated. So it leverages an inflammatory response, right, using macrophages. It can release cytokines to recruit additional inflammatory cells, whether they're neutrophils and or macrophages. If you look at the CD8 positive pathway, which is this right side, it also enters the circulation after it becomes activated. And it goes to the site and it actually kills the infected cell. So this pathway is showing what happens with your CD8 positive cells. And these two pathways are showing what happens with your CD4 positive helper T cells. Remember the CD8 positive cytotoxic T cell, CTLs, that means cytotoxic T lymphocyte. It's going to lyse the cell and kill it. Now I want you to remember, and that's this last bullet point on the bottom left, is this doesn't happen without the presence of co-stimulation. In fact, on the CD8 positive side, there's a unique co-stimulatory event. Because in the beginning of the lecture, we characterized CD4 positive T cells, right? And we characterized the co-stimulation that happens with the CD4 positive T cells. The CD8 have their own, and it looks like this. It uses IL-2. So this little diagram that's blown out, this is a box diagram that comes in here. Your antigen-presenting cell presents to what we call a naive cytotoxic T cell. That naive cytotoxic T cell releases interleukin-2. IL-2 is a cytokine. It releases interleukin-2 when it recognizes that this presentation of a non-self antigen is going to want to trigger a response. Interleukin-2 through a autocrine response binds to the same cell on a receptor, and this autocrine activity is called co-stimulation. So that's co-stimulation using IL-2 in an autocrine fashion for CD8 type of lymphocytes. Did you catch all that? Can we say it one more time? Okay. CD8 positive T lymphocytes, when they're naive, they're exposed to an antigen. The antigen is non-self. They release IL-2. IL-2 binds to a receptor on that same cell. That would be considered an autocrine response. That causes the co-stimulatory event for that cytotoxic T lymphocyte. Now it's activated. As it becomes activated, it manufactures effector T cells that are going to go kill, and it manufactures memory T cells to store that, similarly to what you do with your B cell. Okay? So the memory response comes from the cytotoxic T lymphocytes after a co-stimulation event of IL-2. So the cytolysis, there's some more details here of how it lyses the cell. And there's two main mechanisms that CD8 cells use. They're called cytotoxic T lymphocytes. This picture is just that previous picture blown up on this side right here. So this is the activity of the CD8 positive T cell. Well, the infected cell with microbes in its cytoplasm is going to go out, right? It's going to be discovered by the CD8 positive T lymphocyte. And how does the killing take place? Well, one of two mechanisms. Either an extrinsic pathway or an intrinsic pathway. Now, it's interesting because we call it cytolysis. It lyses the cell, but it's really an apoptotic mechanism that they use. So if you look at both of these pathways, they both end in apoptosis. Cellular lysis, you would think, would be more of a necrotic pathway. So they go kill, but they do it in a very organized fashion. Now, the details are less important. The extrinsic pathway binds to a perforin plus a granzyme, which is a receptor, and that triggers intracellular caspases to cause apoptosis. The intrinsic pathway utilizes a receptor on the inside after activation called fast cell binding to FAS, and that triggers an alternative trigger pathway of apoptosis. But the most important thing to remember, don't memorize, I would say, all of the details, I would say that no, the cytolysis that takes place either is extrinsically or intrinsically executed, and they both lead to apoptosis. So it's not a mess. It's a very organized killing of those particular infected cells. But that should make sense to you. If you have a cell that's infected with a microbe, would you just want to go there and start slicing and dicing and spill all of that bacteria all over the place? No, you want to come in there, trigger it, and say, let's shut this thing down and actually compartmentalize and clean up this mess. So it actually does make cellular sense. Okay? So it's really more of an apoptotic mechanism than it is a necrotic, ditching everything all over the place. All right, lastly on T cells, the memory T cells. Once they become experienced, and they become experienced by a previous antigen exposure, we call that sometimes an antigen-experienced T cell. So on the second encounter, they mount a faster and a stronger immune response. So how does this happen? You can see a different way of looking at it from this paper that I pulled. You have a naive T cell, and upon stimulation, right, it's exposed to the antigen, and IL-2 is released, and IL-2 binds on its same cell, on an IL-2 receptor. You get a population of effector cells. Those effector cells can actually go trigger this apoptotic signal that we talked about, and they later on become the memory cells that circulate in the tissue, and memory cells that actually circulate in the lymph node. So we actually have two populations of memory cells, one that hang out in the lymph node, and one that hang out in the tissue. So the tissue where it found it, it leaves soldiers and leaves a camp and says, stay here. And then it makes a secondary population of lymph node homing cells to say, you might find it circulating back through the lymph, so let's hang out a population within the lymph itself as well. Okay. So as we wrap up for today, let's go through a couple of questions to kind of gear you up for the exam as it relates to this section. Which of the following is not a correct pairing? CD4 positive helper T cell, CD8 positive MHC class 1, CD4 positive MHC class 2, plasma cell antibody, APC fibroblast. How many say A? B? C? D? Or E? Okay, the E's have it. Why? Why do the E's have it? The fibroblast is not traditionally considered an antigen-presenting cell. Okay? That's good. Is it a somatic cell? Yeah. If it was infected, it could present an antigen? Technically, yes. But it's not classified as an antigen-presenting cell normally. Okay? The other matches are actually correct. Okay? Plasma cells and B cells are the ones that manufacture antibodies and produce antibodies. Okay? All right, next question. After degranulation, that means they dump their granules, mast cells release prostaglandins and leukotrienes. These perform which of the following functions? Vasodilation and increased vascular permeability. Attraction of CD8 cells. That should be CD8 positive cells. Activation of the complement cascade. Opsonization of bacteria. Or three of the above. Who knows the answer and why? Are you fixing your eye or hair, or you have the answer? Yeah. Okay, you're itching your eye. No answers? We have a test coming up. You can shout out the answer, but you got to be able to defend it. Just kidding. So the answer here is A. Vasodilation, increased vascular permeability. Why?
What if it's due to inflammation and that could cause all the branches of the blood vessels and all that stuff
you guys hearing that? She says, isn't this due to inflammation? And the answer is absolutely. Okay, mast cells degranulate to release prostaglandins and leukotrienes. You remember, those are products that are found in these granules that fuse with the membrane, right? They're manufactured from what? Where do you get prostaglandins and leukotrienes from? How do you make them? Arachidonic acid that comes from where? The phospholipid bilayer. Do you remember this pathway one? You generate prostaglandins, leukotrienes, thromboxanes, and prostacyclines. You remember those 4? I would review that if I was you. That would be good to know. But yes, you're absolutely correct that this is because of inflammation that this process is happening. Okay, another review. Which type of necrosis do you see as a result of glomerulonephritis? Now, we didn't cover glomerulonephritis yet because we haven't covered renal. Bless you. But what is glomerulonephritis attacking? What organ? Kidney. And so, because you know what organ it is it coagulative, caseous, fat, gangrenous, or liquefaction? What is it? Coagulative. Why is it coagulative? Pardon? Because it is? That's the type of necrosis. Why is it coagulative? What characterizes coagulative? What makes coagulative have the characteristics that it has? What's that? I can barely hear you.
Like the glomerulus?
The glomerulus?
Yeah.
Okay. Yes, because of the vascular architecture. Remember, organs that have a branching-like pattern are going to have a wedge-shaped necrotic lesion. So we have kidneys, spleen, right? Heart, all of these actually, liver, they all would be a coagulative type of necrosis. So you only really need to know the organ to know if it's coagulative or not. Okay, here's another one. Is this helping? Yes. Or scaring you? Or all of the above? Or A and B only? In an experiment, bacterial organisms are added to a solution containing leukocytes. Engulfment and phagocytosis of microbes happens. Then you add something that enhances the engulfment. Which of the following substances is most likely to produce the observed effect? A. Complement C3b, glutathione peroxidase, immunoglobulin E, P-selectin, or NADPH oxidase. What's the answer and why? A? A is correct. Why? It stimulates opsonization. That's exactly what it does. Nicely done. All right. This is the last one, and then I will let you relax. A 9-year-old girl sustains a small 0.5 centimeter long laceration to her right index finger while playing Queen of Swords with a letter opener. Which of the following substances on contact with the injured vascular basement membrane activates both the coagulation sequence and the kinin system as an initial response to this injury? Thromboxane, plasmin, PAF, otherwise known as platelet activating factor, Hagman factor, or histamine. What's the answer and why? What's the question asking? I don't know. That's a good question. I want to know a factor, right, or a molecule that simultaneously could activate coagulation and kinin systems. That's what it's asking. Queen of Swords, letter opener, those are all just distractors, right? Although it happens in my house. Distractions do happen. But they help with great test questions.
Is it C?
C? Close. What's that?
Is it also intrinsic factor?
PAF?
Yeah.
No, it's not C. but you're on the right track. It's actually D, which is Hageman factor. This is another one of those slides in the slide deck sequence where we were talking about coagulation and the kinin pathway, and we're looking at the cascade of events leading down to thrombosis and Hageman factor triggered both pathways. Okay? So look back at that slide as well. Any other questions? All right.